you can understand the status of artificial intelligence. Yeah. So now, seriously, sorry for this small interruption, but this computer is used every week for 2 lectures, so something about this is very strange. So I'm really pleased and happy and honored to be here. And I forgot what I wanted to start because of this, but now. So you will see the presentation, which is radically different with respect to the first one. So it's first stating what we really can do, because we had yesterday a lot of discussions with peers about what will be done in human time, and we made some bet with Marcello. We are ready to make it official, and so I'm just trying here to follow up on the questions we made in the first philosophical lecture into what really happens today at the standard of care, both regarding implants and regarding the use of artificial intelligence in implants, because there is too much hype, and let's diminish that hype by today. Then tomorrow maybe something next will happen. So to do so, I will guide you through, again, inspired by yesterday beer, so do the conference dinners before, one application of invasive, one application of non-invasive that we've been developing since a while, and one is regarding the amputees, the other is regarding the diabetics. So let's go on a journey. So what are the specific challenges in this multidisciplinary research field where we are working in? First, we are trying to face the clinical unmet need of aging population, in this particular case like diabetics and amputees. And what you can see here is a real walking of severe diabetic patients with a neuropathy. He needs assistance, he's very slow and asymmetric. And alongside with this low mobility, possible falls, and especially pain, and there was discussion about the pain before, they importantly lose the independence. In my lab and in overall in our Center for Neuroprosthetics, we are trying to communicate directly with the nervous system. To do so, we have to understand both which devices to use in order to communicate, so how to interface them, and also we have to understand how to speak to the nerve. So what is the policy of stimulation in order to be speaking on any of the things that have been mentioned before. As you're gonna understand in a few slides, we are very limited there. And discussions about changing personality of humans are probably decades away. So regarding those neurotech solutions, afterwards we really need to think about them. We need to think about the pragmatic facts, also how to place them into the patients. We need to go to the cadavers. We need to try the placement not of the devices, of the cables and all the system. And finally, those devices potentially can restore the lost sensory motor function or help us with the pain management. So how does it apply to a very important thing, which is again philosophy, but research philosophy in this field? So we first start from a deep understanding of the system through the development of biomimetic approaches, and I'm going to show a couple of those. So we try to understand what are we stimulating, or where from are we recording from mechanistic viewpoint. Then we try to make the devices, the technological solutions that are able to fulfill those aims. And then we go to the clinics and we assess directly with the meticulously projected assessments what is that technology doing to the patients. But the real science, the real philosophy of science is not happening here when we publish Nature Medicine and beautiful things and we were invited. It's happening here when we go feedback and then we learn what did we assumed wrong and what has to be changed in the starting hypothesis. And this presentation is a lot about that. So what did we learn from our human implants and what are we doing better with respect to the first generation? So, as promised, I'm following. So the first thing is, how do we speak to the nerves? And I don't want to bother you with the details, but the philosophy is important. So we started from mathematical models that are resembling the way that we feel. So don't bother yourself with equations, but there are some parameters that are resembling the realistic behavior of skin and of the afferents under the skin. And then we place them in a proper way within the foot sole, and then we did experiments with a colleague from Canada where we've been placing this small robot over different areas of the foot sole and measuring to the single neural recording the activity of afferents when you stimulate. And this is important. So again, what is physical with respect what is the philosophy? So to measure one single or two afferents, you need hours-long experiment. So we reassembled many experiments of microneurography, hundreds of them, so hundreds of days of work of humans to measure hundreds of microneurography measured afferent units to be able to fit our model and to make in silico model of the walking. And so that's the first artificial thing here. So we made in silico reality that we can then probe and use in order to make the better neuroprosthesis. The next step was starting from that pseudo-realistic in silico artificial foot, we were probing it with the mechanical stimuli. And so this is what dynamically happens when you walk. So the areas of those afferents are changing. And today, humans, we cannot measure this. We do not know if this is really true. We can only do it with this type of models. Why? Because it is impossible in humans to measure multiple active sites of neurons while walking. It's physically impossible. So we can do it only in silico. So we did that, and then we compounded that activity into the different classes of afferents, as those you can see there. So we made an artificial code in a way that fast afferents 1 are working, fast afferents 2 are working, slow adapting 1 or 2. And I can give more details about it. But I want just to give you the philosophy. I don't want to bother you with the technical details. So the next step in this careful development of technology to implant into human was then to go to animals, right? And then we performed animal experiments in which we had a really difficult preparation, in which we had a peripheral electrode that would induce those electrical stimuli as those you're seeing there. Then we would measure in a couple of positions into the spinal cord, because first in dorsal root ganglion, and then also in the spinal cord. And you need 20-hour surgery to do that. So why all of that? To see if what we are doing makes at all sense. If these information that we are encoding in the periphery from artificial-like system are really traveling to the brain. And finally, yes, this is happening. You can see schematically how is the thing. The thing from periphery where we stimulate is really in a one-to-one way being transferred towards the brain. So we have some idea how to stimulate, so which language to speak. Then, as promised in the first slides, we need to know which system do we need, what system do we make to help to amputees in this particular case. And so here, you can see how the amputees walk. They are asymmetric, very slow, and they don't believe so much in the artificial leg. So we instrumented it with a sensorized insole, encoders within the knee of the prosthesis, and our system controller, the brain of the system, where artificial intelligence is taking those signals and then injecting it through the carefully designed electrodes from fellows from Freiburg, there are somebody, and stimulating the residual nervous system. And then the nature does the rest. The signal is conveyed through the spinal cord that we saw in animals, going to the brain, and user is confident, is again able to walk in a proper way. Or at least in this beautiful simulation that my friend made for me. So it's nice, right? It's cool, and we can stop it here. But what is the reality? So in the next step, and who is afraid of blood, do not watch, but this is the reality. These are implants, right? So we really designed the implants that went into the amputated nerve in the way that our model said, and then the second step is the physical reality. You need to be careful not only about the implants. By the way, those implants are not harmful to nerves. We have shown that extensively. The nerves are harmful to implants because in the leg, the muscles are very strong. So we had to take a particular care about the cables going towards the implants. And this is what we've been doing. That's the trick to do. In microsurgery, we made a flap. This is like a sandwich of fascia, which blocked the cables not to move while we are moving strongly the leg. Then we transcutaneously routed them, and for 100 days we had a very stable situation. Of course, I had to acknowledge that was a big European collaboration with several industries and clinical centers. And then we took, in this implant part of the story, we took 3 patients with a very disability, very high amputation, fear of fail, and phantom limb pain. And then this is what happened. Very few days after implantation, these are the type of sensations that we are able to elicit into different patients with different active sites of those electrodes. Just to be more precise, the small implants, each of them have 54 active sites. So what do is we stimulate through each of those, injecting the current into the residual nervous system, and the users, whenever they feel something, would stop graphical user interface and say, aha, I feel it here, and just design what do they feel, alongside with the type of sensation. And so this is what you come up with the like artificial implantable device. And when that is connected to the whole system, hopefully you'll see, I'm pressuring the sensor. The green box there is transmitting wirelessly this information to the white thing on the table. That's a neurostimulator. And this is a placebo trial, for instance. And the neurostimulator through the cables passing through the skin is stimulating precisely the nerves in a way that we map them. And you can see that user is really reliably feeling different position in a repeatable way for hours. Similar thing was achieved also regarding the proprioception. So we were able to map the movement of the knee towards the closure of the gastrocnemius medialis muscle. So they would be able both to understand where do we touch them. That's a separate set of experiments. There are videos in the paper. How much is closed the leg and where do we touch? And that's not trivial even for us sometimes. So next thing was we ask ourselves, okay, what's there happening? So it may be that we are just disturbing them, giving some electricity which is wrong. So we did a meticulous test in order to understand how do they, and that's coming up to the previous lecture, about how do they feel that, how do they embody it. So in the first set of experiments, we did what we call the subjective embodiment. It is well described in the literature, in which we made the questionnaires, asking them in a proper way how do they feel the artificial device with the neurofeedback with respect to commercial device, and also, of course, some placebo questions. But also, we did this experimental test, which where you see the pool. They cannot see it because they are blocked from seeing it. That they should say stop in a moment that they feel that the pool is aligned with their artificial leg. And what happens is that when they're using the prosthesis like a commercial one, there is a stronger displacement with respect to the situation in which we are constantly stimulating them, because then they feel aligned, the artificial leg, with respect to that pool. That's the first set of the first set of experiments in which we realized that they feel prosthesis is a part of the other body. But in other set of experiments, we went even further. We used electroencephalography, EEG, in order to measure how do they feel about that completely objectively, not by saying, so it's an objective measure. In those set of experiments, they were wearing the headsets, and they were instructed to count some type of tones, like low tones with respect to the high tones. That's well described in literature. It's called the double task. It's used in many other also clinical physiology experiments. And so while they were doing that, they had to concentrate to understand which type of sound is it. And meanwhile, they would be walking either with the prosthesis with neurofeedback or with a commercial prosthesis. And in a third situation, they were just sitting and doing that task. Long story very short, in those experiments, what you see in blue and red is them sitting or walking with a neurofeedback. And black line is proposing what happens when they are working with commercial-like prosthesis. What does it mean? It means that through the ERPs, we are able to measure that they are having much more mental layover. So they are much more relaxed when they have a neurofeedback. It's more, they're more confident, and they could answer to higher cognitive questions that come up again to the previous lectures. Where do we stand? Like, even in so easy actions, they cannot walk easily and count the tones. Imagine something more sophisticated, right? Instead, with the neurofeedback, they were able, and so we had also objective measure that we are doing something well to those individuals. Then in the next set of experiments, we were doing first experiments with obstacles in which patients were walking with some glasses blocking their sight, and they were trying to go over obstacles. And what we observed that neurofeedback is statistically much better when they are trying to go with it with respect to the commercial prosthesis. And in another set of experiments, this very simple task as a stair mobility is very problematic for the patients that have a commercial prosthesis. Instead, with a simple acquisition of only 2 sensors, they are able to walk more confident and more quickly with the neuroprosthesis on. In reality, we also categorized why is that happening, and it's simple. They really learn to have the confidence how they should roll over the leg, and therefore they push more quickly. And the mobility is the key feature for the patients with a prosthesis, because they are risking 200% higher probability to get a heart attack because they're not moving enough, for instance. Last but not the least, in these functional tests, we also ask ourselves, how do they feel the weight of prosthesis? Because despite the modern prosthesis are the half weight of our normal leg, they're always saying that the prosthesis is too heavy. And so we did a set of neurophysiological measures along with the psychosocial scales in order to understand what happens when they use the prosthesis with feedback or without feedback. Long story very short, they felt prosthesis 25% less heavy when they were using a neurofeedback. And of course, again, we measured both the cognitive load and embodiment. And these were the reasons why they felt it less heavy. And now we come to one of the important points. So all of this is nice, functional, but unfortunately our societies are not going to pay for these prostheses like this. It will be aesthetic thing or only functional thing. So we of course want to go towards the end user. We want to understand where can we go and how can we justify future widespread use of those devices. So we must find the tangible health benefits, and we did so. So in a separate set of experiments, we were understanding how the phantom limb pain is going, and on the left is phantom limb pain prior to implantation. The pink areas when we stimulate them extensively during the trial, and after the explantation, after 100 days, the phantom limb came back. And we could robustly show that we are diminishing the phantom limb pain. Indeed, there are 2 companies now that are trying to exploit this technology into the widespread use. They got FDA approval and whatever. On the other side, we also ask ourselves, how are they breathing? Because as I said, since they are asymmetrically walking, they are using too much oxygen, and therefore they have 200% higher probability to get a heart attack. we were able to diminish their oxygen consumption significantly, especially in these outdoor conditions, which gives hope that we would elongate their cardiovascular life. And those things are the one that National Health Sanitary System are going to take into account if wanting to take into the consideration these type of devices. Last but not least, since we are scientists, we ask ourselves what is there happening really? And I'm coming back again because we dream the television is speaking about super fancy codes, everything is happening. But in reality, in our experiments, the users were using quite simple strategies. They would really resemble those artificial sensations into their body. So when they're walking over ground, they would stop rolling like a pirate of Caribs, like pushing that. They're starting rolling because they felt also the forefoot. While going up and down the stairs, they were always able to understand, okay, I feel my heel, I can push, or when they're going up, I feel these three sensors, I can push. And we statistically showed that. So the strategies they resembled were very simple, but were very robust. Because, for instance, if sometimes casually we would turn off the stimulation, they would stop and tell, oh, I lost my knee, or I lost my foot. So after that, we were kind of happy, and the users were happy, but this is where, as I promised, the real science starts. So we come up with understanding what happens worldwide because we made like 10 of those implants, but worldwide, especially in America, there are almost 100 patients with different type of implants. So we summarized all of them in this study, and we try to understand what is wrong with what we are doing, what should be done better. And there are several things, among which the first one, when we stimulate the nervous system, it is often not natural. These white areas and also yellowish areas are representing sensations that users do not love. And you will not find so much literature about that because nobody likes to say that, right? The same thing applies to DBS and especially to spinal cord stimulation. So it's not easy to find that spinal cord stimulation is mainly paresthesia or that many of these are mainly paresthesia. And paresthesia, for whoever is not knowing, is like pressing your finger into the power supply. And you would not love to have that by walking, right? So that's one problem. And the second problem are all these things which you can't see in the very last line, but they are described in this Nature Materials paper in which the thresholds and the way of stability of these devices is very variable from site to site, from country to country. So we wanted to understand more about these things. And so what did we do was we were lucky enough to take the samples from our amputees because this is a residual part of the tissue where they were implanted. And so when we were exporting the electrodes, we could export also that tissue. And then we were able to reconstruct from those tissue samples the realistic structure, 3D, of what is happening or what was happening during our implantation time. So we can understand where are the fascicles, where is fibrotic tissue, and especially where the electrodes were. And so some of nice things that we have observed. So these are 3 active sites, for instance, and this term of stability and instability is very important in brain, in periphery, and wherever. We can have behaviors where the active site from first to 100 day of stimulation is always stimulating the same position, or the third one when the position is very much changing. And so previously there were stories that it is all central plasticity and philosophies about that. But in reality, we have shown robustly through our modeling that it is simply when the fibrotic tissue is growing, and this is one active site, that active site is being pushed from a single fascicle to the fascicle on the other side, and this is why we are changing the position of the stimulation. That's one powerful thing that we have observed. Second, and this applies also to the central implants, we were able to really characterize the fibrotic tissue transfer to these implants. And that's important, and we do not have so many evidences in the literature because there is very few data of explanted from human tissues. And so what you can see here on the left, it's the formation of capsula, the fibrotic capsula around the electrode. This is polyamide electrode, this is capsula, and these are higher and higher zoom in of that. And we could understand that there are 3 types of behavior. There are active sites that are very stable, active sites that are half stable, and there are active sites that are very unstable in those implants. And we could also computationally predict what happens. So just remember this graph. Finally, we could make some outlook about these bioelectronic interfaces. First, we need more simple implants because our surgeries were too long. Second, we need a way to diminish the fibrotic transfer, and that's the holy grail of many of the implant industries. But third, and very important, since we learned those relations that I've shown, we can use, these are really good candidates to use artificial intelligence. And to do so, we constructed some artificial models in order to counteract those actions. And so, first, and don't be afraid from a name, it's called Bayesian optimization. We first try to understand how are we able to deal with this location migration and threshold changes which we have observed during our trials, right? And Automatized approaches, those that Bayesian optimization is offering, are the best suitable candidates to do that. And in very simple words, Bayesian optimization is trying to make the interaction between input and output through the known physical behavior. And that's what we learn in our tissues. So we were able to propose the optimizer, and just concentrate on this part, that is able to go over the line of the stimulation from day to day and to exploit the new points of the stimulation automatically, and to understand when, for instance, users were not feeling anymore the sensation, that the moment is to increase, or when they felt the pain, to decrease the stimulation that was presented. In a separate set of the same usage of the controller, without too much mathematics, we were able to stabilize the active site. So when some active sites was dying, our artificial intelligence-based agent was able to understand which one is the candidate to take over it and to stimulate again instead of the one that failed. That's called exploration, right? Exploitation is using those that work. And both of those things were stably possible. Why? Because we have a redundant number of active sites. We had 56 active sites and we needed 4 of them. But we were losing active sites. So we were able to use those that remain safe in order to stimulate the function. And as I promised to Marcello, because Marcello asked me about invasive versus non-invasive, so I'm giving you an example. What happens when you try to use a very similar technology over non-invasive? So diabetic patients have important issue connected to sensory loss, which are kind of similar to amputees. As we know, diabetes is completely very difficult and poorly understood disease with a complex metabolic interactions, but unfortunately it often finishes in neuropathies, which are killing the small mechanoreceptors in our skin. What does it mean? When you walk and you have a rock in your shoe, you do not perceive the rock, and then the rock is making an injury. The injury over time is recurrent injury, and finally the ulcer. And the ulcers, as many doctors know, are guiding to the amputation and very many death outcomes. And this is what happens in diabetes, but at the same time, they also suffer the neuropathic pain, which is very similar to the phantom limb pain of amputees. So without painkillers, they would not be able to sleep or perform everyday activities. And that's the guy from the starting. That's really a patient with a diabetic neuropathy. And so here we have a trade-off between benefits and invasiveness. So should we implant those users that have diabetes with a similar type of implants which show the great performance in amputees, or should we try something less invasive in order to counteract their health status? So we come up with a non-invasive solution which is trying to break this vicious cycle, because persons having diabetic neuropathy, they are walking less and less, they are falling more, they are earning the weight, and therefore they are walking the less. So we want to break this vicious cycle by diminishing the pain and increasing their mobility, because that would be the life changer for those persons. To do so, in collaboration with Balgris Hospital in Unispital Zurich, we made this device, which is similar as the one for amputees, but completely non-invasive, in which we have sensorized insole, outside system for the stimulation, and purposely designed sock, which is able to target the nerves with the multiple active sites from outside, searching the part of the nerve which is healthy yet. So proximal part of the nerve is yet healthy, while the distal part of the nerve close to the foot sole is getting to the worst condition. And then what we do is quite simple. So we are trying to do the stimulation of the peroneal, sural, and tibial nerve to be able to restore this type of sensation. As you can observe, those sensations are much bigger, much less sophisticated as one with implants, but maybe, and hopefully, in this case, are healthy enough, are enough for the rehabilitation of those patients. So, as said, we did a purposely designed sock, and this is 3 years of engineering effort, and again, artificial intelligence. Since there are many active sites that we can stimulate through, we must have a knowledge which one to use. Because if you would sit and try to stimulate these 48 active sites, that would be hours every day. So what did we do? We did, in this case, the reinforcement learning algorithm. In few words, it's an algorithm that is able, through its agent, which is the brain, to stimulate different active sites, it's a probing of the system, understand the behavior of environment, which is our damaged nerve, understand those, which you can see here, the reward. So the high reward would be, it's a comfortable sensation and I really feel it under my foot. Low reward would be, it's a strong paresthesia and I feel it in loco. And then this system automatically is able to adapt and to understand which set of active sites are to be stimulated to have a reliable sensations underfoot with a minimum in loco sensation and with maximal pleasantness for patients. And when we compare the behavior of reinforcement learning in all of these graphs with respect to the most experienced therapists from our lab and with the more stupid algorithms, reinforcement learning was always performing. And that's a great result. This is where we stand with artificial intelligence. We are able to automatically calibrate that system. And what does it mean? It means you have it here, the time and the number of stimulation. The time that reinforcement learning is taking is 5 minutes with respect to like 25 minutes that naive therapist is doing. And this is the real engagement of artificial intelligence in Perbe, good things done with these really neuroprosthetic systems. And so how does this transfer to the patients? Because we also had patients here. This is one of our very first patients. He's being touched on a forefoot, and he's unable to feel it. Then he's touched over the heel. It's a severe neuropathy. He's like 20 years into diabetes. Of course, we have placebos because he doesn't feel it. May be that now he starts feeling something, And again, we must be sure about placebo effect, right? And that situation expanded to many patients. We had 14 patients in which we were able to restore up to 1/4 of the lost sensations, which they didn't feel due to the diabetic neuropathy. And when we ask ourselves, and that's one of the very final results, how they transfer to the functional, you remember, I said they're slow, they're falling, they're not walking enough. So what we have observed, a specific category of those patients, those with acceptable BMI, so that are not really severe, like almost not to go from home, improved their cadency, so they were walking much better, statistically better during clinical trials, so they were both able to walk more efficiently, but also through the objective clinical measures, which I can explain you during the cocktail, we were able to assess that their overall health situation is getting better. So it's not white and black, but it's like some inclusion group can be treated properly with this. And last but not the least, regarding this, I want to share with you experience of this lady, if it works.
Gemacht, mit und ohne Stimulation zu laufen. Und ich muss sagen, das geradeauslaufen, das einfache geradeauslaufen, was mir schwer fällt, weil ich immer so die Beine so ein bisschen unkoordiniert bewege, ist mir mit der Stimulation viel leichter gefallen. Ich habe auch das Gefühl, ich bin viel gerader gegangen. Ich kann es nicht erklären, warum.
And that is important because she cannot explain why, because we produced a system that is intuitive, she doesn't have to learn. And that's a typical question I'm waiting, how much did they train? And said that, I just want to wrap up that I do believe that what I've shown in an invasive and non-invasive way are the way to make these neurotechs into the society. We changed the model from trying and error. We were in silico model guided and artificial intelligence to optimize it. We passed through the animals. We made these realistic models. Then we knew how to do that. And just at the end, we jumped into the first-in-man solutions that, to my understanding and belief, are enabling healthy aging and meaningful life of those persons with these disabilities. And that's a proper use of neurotech and AI for a concrete purpose. And that's what was about enhancement before. I strongly believe we must have a clearly defined need and fulfill that need, not go around and make some enhancement without some clear reasoning behind. And then I really have to thank all of these beautiful persons that really did this work, and to the multiple funding agencies. Thank you very much.
Well, thank you very much, Stani, for this fascinating talk. It's always great to listen to you because you are really at the forefront at the patient. And I think one thing that we learned and can see here is that there's a lot of trial and error, and you have to really go back and forth, sometimes for years, to make progress. And well, I admire your work, and unfortunately we don't have that much time for questions, but maybe one or two questions, and Niels Bierbaumer has one.
Can you elaborate, because it was quite fast, but good. How did you do the neurofeedback when you did this increase of cognitive sensitivity in the amputees? I mean, you gave them feedback of the P300, or you gave them the EEG average feedback, so you got a slow wave activity, and by that they were kind of better in learning that, or how do we be? You know that neurofeedback in itself has no real cognitive effect, but in that combination, it may have it, or it obviously had one.
Yeah, so neurofeedback was given through the implants, and it was a stable map that they felt all the time. So they would feel a foot heel absolutely proportional with the pressure, forefoot proportional with the pressure, and they would have that. Then we would just measure during the double task the ERPs, and we would do the trigger averaging with respect to the cases in which they're having a neurofeedback with respect to not, and the sounds they have to count. And then this is where we are plotting the ERPs with respect to those tasks. And when you can see is directly the layover, and that's a typical way that it's done for the double task, oddball task, to be more precise.
All right, we have one more question. Kostas.
Thank you, Stan. I want to ask one question with regards to this concept of feedback, because I think that features very centrally in all of what you have discovered, published, and communicated to us. I see that there's a strong subjectivity with regards to feedback on which you're based on. Taking into consideration the fact that these new generation implantable devices are gonna feature closed loop technologies, the whole remit there is to somehow objectify, so remove the subjectivity from the feedback loop. What do you think about that in both peripheral and central applications? So if you do not, for example, have the subjective element from the patient to give you feedback, if you're objectively, if the device is giving you the feedback, would you think that you're compromising the information you're getting, and would that lead to a different type of sensation and feeling from the patients and ultimately efficacy?
Oh, that's a great question, and it's half an hour answer, but just to make it very to the point. So first, they do a lot of experiments with monkeys, non-human primates, with the double force choices tasks, where they try to objectivize. But then the ethical question and my direct question is, we do not know if those monkeys are feeling one pain and one in comfort. We do not want that, right? That's the first. Second, yes, we do in humans, so we are happy and lucky enough that we can have a pseudo-objective or quasi-objective type of answer, which we are forcing. So not only us, there are several published papers about the tables that are to be used both in a central neuroprosthetics and in peripheral neuroprosthetics. And we are using very similar scales. So we are enabling users to choose, I feel touch, but also tingling, also spider, but we are disabling them to describe completely novel sensation, like the white, yellow light is going through my nerves. That's the first. Second, the task is those through the brain measurements, through the embodiment measurements, or the quantitative task in which you randomly change the situation with neurofeedback with respect to not having it, are the most objective and functional ways of measuring if it is useful and in any way. So long story very short, there are already several levels of toolbox, subjective, semi-subjective, and very objective, that can measure it. Last but not the least, very interesting results are found now in the brain. You ask about the brain, because we are collaborating with Chicago about those aspects as well. They have observed a lot of interesting results about perception of sensation in the first human subjects with brain implants, right? And so for me, it's very surprising. They often feel very pleasant sensation. And a priori, we wouldn't tell that, right? So experimental knowledge and the feedback from users is essential. And just to mention, they have a pleasant sensation, but they don't have a strong modulation. So whenever you increase the stimulation, they have a nice touch on the tip, then they lose it, or they have very strong touch. So there are pros and cons of all approaches, but we must include human into the loop of design of these type of devices.